Hello everyone, welcome. I would like to talk about DNA discovery today, but I want to make it very short and sweet. So please pay attention, please be tuned till the end. I'm going to try to accumulate everything and put it in a very short video. All right, so let's get started. All right, so DNA discovery, let's talk about the timeline. So it was Johannes Mescher in 1869 who discovered about DNA. He said that DNA is rich in nitrogen and phosphorus, and it's not a protein. However, he did not find out the functions of DNA. So that was further carried out by Griffith in his experiment after 16 years to find out the function of DNA, which was further confirmed in 1944 by Emery, McLeod, and McCarty, and it was further confirmed by Hershey and Chase, whether DNA is the genetic material or not. So let's talk about the experiment that was done by Griffith. What did he do in his experiment? So he was working on pneumonia bacteria, the bacteria that causes pneumonia. So he took out, there are two strains of that bacteria. One is the S strain and one is the R strain. So out of these two strains, the S strain, sorry, fancy, the S strain is considered to be virulent or pathogenic, means it can cause pneumonia. So this is considered to be pathogenic or virulent, but the R cells or the R strain is considered to be non-pathogenic or non-virulent. Okay. This is S. This is also called the smooth strain, and R is also called the rough strain. So what happened? He injected the living S cells into the mice, and he found that the mice died. Of course, because they were virulent, they were pathogenic, so mice died. Then he injected the living R cells into the mice and found that the mice lived. Obviously, they were non-pathogenic, so nothing happened to the mice and they lived. Then he injected the heat-killed S cells. They were killed, so they were heat-killed S cells were injected into the mice, and they found that mice died. And in the final one, there was a surprising result. When he mixed the heat-killed S cells, they are heat-killed, with the live R cells. R cells are non-pathogenic, right? And the S cells, they are heat-killed. But then when he injected this mixture into the mice, he found that mice died. Now, what was the reason why mice died here? That was the whole question. So in that case, the further experiment was carried out by Avery McLeod and McCarty to find out if DNA was the genetic material or not. So what did they do was, they took out from the S cell, or you can say from the bacteria, the heat kill S cell, they separated the components of the heat kill S cell. So they took out in three tubes the components of the S cell and added DNAs to the first one, RNAs to the second one, and protease to the third one. So they already knew that the cell consists of DNA, RNA, and proteins. This part was already known by them. So they took out the three tubes, separated the components, and added these three enzymes in them, DNAs. Once DNA is added means that the DNA will die. No more DNA is there. So this tube had just RNA and proteins. Now another tube was subjected to RNAs, means no RNAs there, so it had DNA and proteins. The third tube had protease in it, means no protein. These enzymes kill their respective material. So protease will act on protein, so no protein was there, only DNA and RNA was there. All right. So then they mixed this mixture with the R cells, the living R cells. And what was the result they found? Depending on that, they found the answer. So then they mixed this mixture with the R cells, the living R cells, and found that the mice lives. When they mixed this mixture with the R cells, found mice dies. And when they mixed this mixture with the R cells, they found that the mice dies again. So what happened in this one, in these two cases where the mice dies, and this one the mice lives? So here in this case, the RNAs was there, so there was no RNA. There was no RNA. The DNA and protein was there. DNA and protein is there. When this is mixed with R cells, it means something is being transformed in the R cells. It means that the DNA from this or the protein might be moving from these cells, from the S cells to the R cells. Might be. So DNA or protein is moving to the R cells and it is transforming the R cells into the S cells, which are virulent or pathogenic. And that's why mice dies. In the second, the next tube where they added protease, now it has DNA plus RNA. It could be the same thing. When this is mixed with the R cells, it could be that DNA or the RNA is changing the components in the R cells to S cells and becoming virulent. So that's why the mice dies. But in this case, it was found when the mice lives. So only DNA is added. So no DNA is there now. Only RNA and protein is there. So when RNA and protein are there, none of them are able to transform the R cells into the S cells. None of them are able to convert this into this, therefore the mice lives. So it was automatically proved that the DNA is the genetic material. DNA is the transforming principle. So in this experiment, it was like, what is the transforming principle? Why does this mice die? What transformed the R cells into the S cells? What went from S cell into the R cells and made them virulent? So it was found in this experiment that it was DNA, which is a genetic material, which went from the S cells to R cells and made them pathogenic or made them virulent. All right. So this was all the short, concise part of the DNA discovery. I will cover Hershey and Chase experiment in the next video. All right. Thank you. Bye-bye.